Good evening, everyone. Thank you so much for coming and welcome to tonight's talk, Antimicrobial Resistance, Diagnostics for the Future. Antimicrobial resistance is an issue that the World Health Organization have described as one of the top 10 global public health threats facing humanity. In this talk, Dr. Robert Hammond will discuss his work on the part he plays to battle this threat and his award-winning machines. What is the theory behind this technology and why is it so important? So a bit about Robert. Dr. Hammond is an accredited biomedical scientist with a particular interest in microbiology. He came to St Andrews Medical School to do his PhD in 2012, having done his undergraduate and masters at the Universities of Strathclyde and Glasgow, where he discovered his deep fascination for microorganisms generally, and in particular, those that cause disease. In 2016, as he began his postdoctoral training, Robert and colleagues won a Longitudinal Prize Discovery Award, which was followed in 2018 with the Scottish Life Science Innovation of the Year Award, both for this novel technology. And last year, we're delighted to say he won the Young Engineer Award from the Royal Academy of Engineering. Dr. Hammond is now a lecturer in the Division for Infection and Global Health at our medical school. So welcome, Robert. Thank you very much for being here tonight, and over to you.
Thank you very much, Tanya. That was a very kind introduction. I really appreciate that. And I really appreciate the invitation to speak to you all tonight. I hope I can give an entertaining presentation and a little bit of an insight into the world of engineering, biomedical engineering, of microbiology and what happens behind the scenes in hospitals where I used to work, and what we're trying to do to change things to make things better for everybody and improve health around the world. You will see throughout the presentation, as right now, there's a few little QR codes scattered about. The first one there is for my personal website, which has also got the address on the screen. So if you want to scan that and or just type the website in, you can have a look at that. It's got more information than I'm able to give you in this relatively short presentation this evening. So moving on, it says there AMR, Diagnostics for the Future. AMR is a shorthand that I'll be using throughout to discuss antimicrobial resistance and this really serious problem that Tanya has so well discussed for us already. So there's a quote that I like to start my presentations with that's attributed to Lord Ernest Rutherford. You can plan research, you cannot plan discovery. And this is a perfect quote for my work, because the machine I'm about to discuss with you, one of my best achievements through my career, called the SLIC, the Scattered Light Integrating Collector, was a complete mistake. It was built to do an utterly different job. It wasn't supposed to do what it does currently. But it does what it does now extremely well. So I stand very much by this quote by Lord Rutherford, that you can plan research, you can plan what you want to do in the lab each day, every day, but you can't necessarily plan what you're going to find out when you're there. What is the problem and what is the opportunity? As we said, antibiotic resistance is a seriously pressing problem, and that's understatement of the year so far, I think. Antibiotic resistance has been described variously as a slow motion tsunami by Margaret Chen, who was the old head of the WHO. It's also been described as the antibiotic apocalypse, and returning us to a pre-1940s, that is to say pre-antibiotic era. That's where people undergoing simple procedures had a high, almost 100% chance of catching an infection. Their chance of dying from that infection were incredibly high. And that would mean an end to all elective surgeries, because the risk would just be far too high without antibiotics to help us. We use antibiotics as a prophylaxis. Not many people are aware of that. That's very common. They're also used as treatment, of course. The issue of AMR, antimicrobial resistance, is caused by excessive or inappropriate antibiotic use. It's not the only reason, but it's a large reason. Some people will tell you that it's down to human use, which is partially true. It's also down to use in animals, also down to the use in the environment. There is the One Health initiative being promoted by Scottish and British governments, where we try to include environmental health, animal health, that's both farmyard livestock animals and companion animals, and human health in a single cycle, where we understand that everything is actually brought together and everything is linked together, which is perfectly true. So the use of antibiotics in livestock is a major driving factor of AMR around the world. Another issue with more coming into the lab side of it and the clinical side of it, the speed of progression of infection is much too fast for us to generate results some of the time. Not for all infections, but for one of the case studies I'll show you in a moment, for a blood infection, what variously is called bacteremia or septicemia, the amount of rapidity of infection, how fast infection spreads, is far too fast for the current technology that's out there. Actually, I was just discussing with our hosts before we came on online there. They asked me what's the question. They asked me the question of how rapid is the current technology? The answer to which is far too slow. It takes about 2 days to get a result, but we'll touch upon that a bit more later on. And the cost of equipment and the cost of testing is far too high. The cost of equipment can range from 10,000s to hundreds of thousands of pounds or euros or dollars for a piece of equipment, and the cost of testing, the individual test that is taking place, can also be far too high. So what have I done to try and solve that, and what have me and my team and I done to try and solve that? Well, we have a rapid and cost-effective solution that addresses a global market, and it is global. One of the things I like to do is work in low and middle income countries. I have colleagues throughout the world, and I currently have an ongoing project in Kenya trying to look at urinary tract infections there. Again, we'll touch upon that later on, but my solution is not only easy to use, but it's also very cost effective, very cheap to use and cheap to run. So as I say, the problem solved. What is it we're trying to do? Really, when you come down to it, all of clinical microbiology is boiling down to a fairly simple trick. You're trying to find the bacteria when you're talking about bacteremia, particularly. Of course, we talk about fungemia with fungi, and there's virology as well. But if we focus down on my work with bacteria and fungi, you're trying to find relatively tiny amounts of particles, bacteria, in relatively massive volumes of liquid. And specifically the minimum possible detection time, or TTP, time to positivity, for organisms is really what we want to try and do. So how have we done that? Well, I mentioned the scattered light integrating collector. You'll see another QR code that will take you right to the SLIC page on my website. What is it? Well, it's exactly what it says. It's a scattered light integrating collector. The physics of it are quite interesting, but I'll not go into that this evening. Essentially what it does is to shine a laser through a sample. The light is then scattered, and if there are more things in the way of the laser, the light scatters more. So as bacteria grow, as we'll see in a moment with some graphs I'll show you, as the bacteria grow, there's more of them, there's more particles in the way of the laser, the line goes up. And so that tells you that there's something growing there. Fine, okay, so we've taken a sample from the patient, we put it in my machine, and something is in fact growing. Good to know. What do we need to know next? We need to know its AST, its antimicrobial susceptibility testing profile. What is it resistant to, and what is it susceptible to? Now, many of you will have heard these words before and understand what they mean. If you take a drug, an antibiotic, and put it on a bug on a plate, not in a human being, but in a lab environment, a plate not unlike this one. I happen to be in a lab in Germany just now. And you put some antibiotic on this, some of the bugs will die and some of them won't. And that is its antibiogram, that is its AST, its antimicrobial susceptibility testing. And that's what we're doing. That's essentially what it does. If the bacteria are growing, then the drug didn't work. If the bacteria stopped growing, the drug worked. It's very, very straightforward and very simple. But the trick of SLIC is that it is incredibly sensitive. It was an iterative process to build it. So we have the original version was built out of modeling foam. I don't know if anyone online likes to arrange flowers as a pastime. This is the same stuff you might use to press dried flowers into in a vase to keep them in one place. Exact same stuff with my very first version. The second and third and fourth version, the second and third version, sorry, I should say, were 3D printed. The first one was referred to in my lab by my colleagues affectionately as the Death Star for obvious reasons. That's this one here. You can see, if you can see my cursor, number 2. 3.1 and 3.2, the aluminium coating went on the inside instead. And then we moved to version 4, which was built for us by a contractor, much more sophisticated version. But it only had one channel. You could only do one sample at a time. I can in fact count. I do know that 5 becomes between 4 and 6. Version 5 is not on there because it was a simple, very tiny proof of principle for version 6. But version 6 was important because it was also 6 was an important number. It was also our first multiplex machine. It allowed us to have 6 samples side by side. Now that's very important because that allows you to suddenly take this machine that's a benchtop proof of principle that's quite interesting academically but has limited clinical use, and suddenly I can take a sample from a patient, urine for instance, split it into 6. Don't put an antibiotic in one, that becomes our positive control, and we add antibiotics to the other 5. The 5 most important drugs for a UTI in Britain, in Fife where I work, are amoxicillin, co-amoxiclav, nitrofurantoin, Nilazid, and other drugs. Not important what they are particularly, teicoplanin sometimes as well, but you can now do an antibiogram, a real antibiogram, and that was a few years ago, and that was the machine that won us the Discovery Prize in Longitude group. This is an example of the sensitivity I just mentioned. There is a device here, again, if you can see my cursor, there's a simple spectrophotometer. Basically what it does is to count cells as well. And when compared to SLIC, it is really, really insensitive. The lower limit of detection for a spectrophotometer is about 10 to the 5 or maybe even 10 to the 6 cells. Now, for comparison, 10 to the 6 cells is 1 million cells. It might not seem like a lot, and it's not really, they're very small. But the new guidelines for a UTI, for urinary tract infection, have lowered the limit from 10 to the 5 cells per mil of urine, per milliliter of urine, to 10 to the 4 cells per milliliter of urine. So you can't use a spectrophotometer to diagnose a UTI. It's impossible. It can't see that many cells. It can't see that few cells, I should say. SLIC, on the other hand, with these organisms here, these 6 extremely common UTI organisms, Klebsiella pneumoniae, Proteus mirabilis, E. coli, Staphylococcus aureus, Enterococcus faecium, and Streptococcus saprophyticus. You can see there that the difference in the lines between the SPEC, which is the square lines, and the SLIC, which is the circles. The limit of detection is wildly different, wildly better for SLIC. It is incredibly sensitive, which is really important and really exciting, but it doesn't tell the whole story because that's static. That's just a moment in time. What about doing things dynamically? Well, some of you may have heard of the drug meropenem. It's very, very potent. It's a very good drug, and it kills off E. coli very well, as you can see here. But the important thing from this graph is the x-axis. It's this part here. It's this time up to 30 minutes. The reason that we were able to be awarded the prize from the Longitude group, Nesta, for our Discovery Award, was because they had decided that 30 minutes was the test, was how long a test should take for it to be rapid. Now that's extremely difficult. Bacteria do grow quite quickly, but don't grow quite that quickly. E. coli doubles its population every 20 minutes under ideal circumstances. Under ideal circumstances is important because taking E. coli from a patient, especially from something like a urine or from blood, is not ideal circumstances. So they're going to grow more slowly than that. So we're thinking about one generation, one doubling of the cells, which isn't very much. If you start with 10 to the 5 cells or 10 to the 4 for urine with the new grouping, that's only 1,000 cells. You're looking from 1,000 to 2,000. That's not a big change, really, because they're very, very small. So within that time frame, they asked, can anybody do this? And we put our hands up and said, yes, we can, and we won a prize for it. And this is one of the graphs that won us the prize. The fact that we can see meropenem, an effective drug, having more of an effect as you move up a concentration gradient is really, really important. I can report to a doctor on the ward with a patient who's suffering with a UTI or with someone who has a blood infection, even more importantly, that within 30 minutes, I can say 4 milligrams per liter of meropenem is going to cure your patient. Get them treated. That's a world first. That's impossible with other technology. So that's the importance of SLIC, is if it was on wards today, we'd be saving lives today. So more data there just to show another drug, gentamicin, as opposed to meropenem. And the same effect is occurring, so we don't get this lytic effect. There's not something dropping below the x-axis, but we are still getting a very, very potent effect within 30 minutes. Even when we use very, very tiny changes in the amount of drug, you can still see a significant difference because of the sensitivity of SLIC. We have two case studies I'll run through quickly for you. Both of these were done out of the University of St Andrews with colleagues of mine, both of whom were actually run almost exclusively by an old PhD student, Kerry Falconer, who's moved on to bigger and better things since she worked with me. But we worked in Victoria Hospital in Kirkcaldy. We took 36 samples, really quite a small study, from routine practice, midstream test of urine. If anyone online watching has had to deliver a urine sample, you know you're supposed to do it midstream, not when you first start peeing, but halfway through. It is challenging. The average age of our participants was 56 years, but that ranged from 14 days, so neonate patients, right up to 87 years, 79 of which of our patients were female. Urinary tract infections do affect women more than men, and so it's a really important problem to try and solve, especially in low middle income countries where often the treatment is not available. So that's what we're trying to do. We study in Kirkcaldy anyway. What could we do? Well, we took the urines. We didn't know what organism was necessarily growing in the urines, but we used our panel of drugs here, penicillin, meropenem, moxicillin, gentamicin, and ciprofloxacin, and we found really nice results. What we found was, in this left-hand graph here, that the gentamicin wasn't working at all. That was terrible. This bug was resistant to gentamicin. Perhaps this patient had to take gentamicin for another complaint before this. The amoxicillin was somewhat effective in the center here. That would have worked for this patient. It was okay. But I could say with confidence that the penicillins, the gentamicin and the ciprofloxacin, the amoxicillin and the ciprofloxacin were really, really, really effective. And that's fantastic. We can see there's almost no growth there. Again, within 30 minutes, we can report that to the doctor. He can prescribe the right drug, and this person's going to start getting better straight away. In this case, a much more resistant organism, resistant to absolutely everything on the panel there, with the exception of meropenem. Meropenem in that case was the effective drug. So this was a resistant strain, again within 30 minutes. And this was a resistant strain, much more dangerous, much more difficult to treat for a patient. And it's a very good chance that this patient wouldn't have got the right treatment to begin with. But with SLIC on the line, we could have said meropenem from the beginning, patient gets better immediately. So here is the stats that came out of that test. Conventional testing, 18 positive and 18 negative, so that was great. Sensitivity and specificity were at 100%. PVP means positive predictive value, was 82%, so we predicted the outcome correctly 82% of the time. When it was negative predictive value was 100%, so that's great. We always predicted when a sample was going to be negative. And all false positive results, that's the 82% there, using additional testing, we found actually that when we compare ourselves to the gold standard, what the lab already does, we were actually more sensitive than the current lab techniques. That's why that PVP is not 100%. It should have been in all rights, but we had to compare ourselves to something. Another case study, this is in pre-publication by, as I said, Kerry Falconer. This was at Ninewell Hospital in Dundee. We used 101 samples, much larger sample size in this case, from routine practice of blood. We only use samples that flag positive for gram-negative organisms. That's things like E. coli, Proteus, Klebsiella, and not things like Staphylococcus and Streptococcus. We compared this to distribution assays, which is the standard of care in Ninewells Hospital. And one of the things we found was overwhelmingly the number of infections in the blood was from E. coli. You can see in this graph here. I apologize, the text is quite small, but the first bar here is E. coli with nearly 150 individual cases coming through the lab of E. coli, with very, very, very few down here of the more exotic organisms. Yersinia, the same genus that causes the plague, Yersinia pestis, not the same organism, and some more interesting organisms down here. But then we followed the E. coli by the Klebsiella, Proteus mirabilis, Pseudomonas aeruginosa, Burkholderia, and some other organisms. So that was one of our kind of standout results, that almost all of the blood infections were with E. coli, very important organism most people have heard of. And the results here were actually arguably even better than with the urine study. It was a much smaller study, it's a much larger study, so the stats are more robust. And with the standard workflow, sensitivity was nearly 92%, WOWS is 95%. Specificity for their standard workflow was nearly 100%. We were slightly lower at 98%. PPV was, positive predictive value was 97.8. We were only at 90, but negative predictive value for theirs was 98, and ours was just almost 99. So very, very comparable data there, and very, very high numbers, all in the 90s. And all of these numbers are well within the FDA's approval rating for getting a piece of equipment approved and rated and into clinics to help people. That's all we're trying to do. We're trying to get our machines out there helping people. So all of this data helps us do that. Sensitivity to antibiotics, I mentioned earlier on some of them, the ceftriaxone, meropenem, ciprofloxacin, gentamicin, amoxicillin. As you can see, the numbers here are extremely high. We found every single meropenem case where there was resistance or susceptibility, every single gentamicin case, very, very close to all the ciprofloxacin cases. Ceftriaxone and amoxicillin, comparably, we missed 1 or 2 there, but that's well within range for, again, for all clinical labs. And if we tested twice, I'm sure we would find all 100. It's important to point out that these numbers up in the 90s are better, or at least as good, the current technology being used in hospital labs around the world just now. So SLIC POC, SLIC POC, point of care. What do we want to do with SLIC? Well, we want to answer a simple question, as I alluded to earlier on. You come into the infection, someone online there has got an infection, you come into the hospital and you say, "Doctor, I think I have an infection, can you help me?" The doctor says, "Yes, of course I can, let me take a sample." And there's 2 things that are going to happen. You're either going to get a yes or a no. If it's a no, you shouldn't get an antibiotic. Antibiotics won't help you, and that's what's driving resistance, in getting antibiotics where they're not needed. So that's great. You're not sick. You still have symptoms, something else is happening to you. Doctor does further investigations. However, if there is an infection, the answer is yes. That takes less than 10 minutes with SLIC. We get a yes answer. Susceptibility in about 30 minutes. So that's 40 minutes from the time you've walked in the door and you have your result. And you see there a stack of red plates. What happens to them? Well, they disappear. These red plates I'm working on in my lab in Germany, all gone. We don't need them anymore if we're able to get SLIC out there in the world. This is the surgical context for SLIC. So what I'm actually doing here in Germany is I am working with some esteemed German researchers and scientists and surgeons, and they're bringing me samples right from the operating theatre, fresh as it were. And what I'm doing is I'm running them on my machine. I have one right next to me here. And today we had a first in class, a first ever in the world situation, which I'm very excited to share with you today. This is hot off the press. This has not been shared anywhere else. I will publish this soon, but this happened today. It's very exciting to be able to share it with you. A surgery was ongoing downstairs. The surgeon suspected there might be an infection. They sent a sample to me, obviously to do the normal protocol as well through the hospital, which takes a long time, as I've alluded to, but they sent it to me as well. I tested it. In 30 minutes, I could tell that there was an infection present. I then used some of my antibiotics, which are sitting here in front of me, and I tested that sample, and I found that it was indeed resistant to some of the drugs in front of me, but susceptible to other ones. I then phoned down to the phoned down to the surgeon, excuse me, and told him what I had found. He wasn't allowed to act on my evidence because it's not come from his official protocol. But the most important thing to point out is that while the surgery was still ongoing, when there was a query of an infection, we were able to deduce that there was an infection and how to treat it. That is a first in class, first in the world. That has never happened before. Very, very exciting. And so to go through this little algorithm here, exactly what happened downstairs, suspect material. Is there growth? No. That wasn't the case. We found growth, but if there hadn't been, is it a virus? We need to do further testing because sometimes viral infections can present like bacterial infections. If the answer is simply a no, or we're convinced it's not a virus, no antibiotic, the patient doesn't need an antibiotic, there was some other reason the tissue looked strange. If there is growth, like there was in my machine, it must have been a bacterium. We send it for MALDI-TOF and identification. MALDI-TOF is a way of identifying bacteria very rapidly using a mass spectrometer. I then did my susceptibility test. We then were able to issue rational therapy, that is as opposed to empirical therapy, which is essentially very, very calculated guesswork by doctors who are highly trained to get it right most of the time, but sometimes they don't. And if we're able to give rational therapy, we know it's right every single time. And it says 2.2 hours there. That was me being conservative when I first wrote this slide. Now I know it's more like, certainly under an hour, more like 45 minutes, as the case was today. So what are the applications of SLIC? There are a great many. Quality assurance in many different places, medical and veterinary. I'm moving into the veterinary space as well. One of my degrees is in veterinary microbiology. Any device, any liquid that needs to stay pure, that is not full of organisms that are growing. Cosmetics, comestibles, water purity in the environment, all possible. Combined with other technology, there are things in labs called flow cytometers, nephelometers, and microfluidic technologies. We can combine our base technology with all of those to improve their sensitivity. But the other applications, or the most important applications, as I've alluded to multiple times, because it's very important to me, is it's cheap. The machine is cheap. It's easy to use. It's easy to use, and it doesn't cost much to buy or to run. So low and middle income countries that don't necessarily have access, or not always have access in rural areas, for instance, they can take one of these machines or several of these machines for not very much money, and they can have access to the highest quality of AST, antimicrobial susceptibility testing, that's currently available. We have a paper out on the SLIC machine. A simple label-free method reveals bacterial growth dynamics and antibiotic action in real time. Highfalutin title, it's in Scientific Reports from the Nature Stable, and that QR code there will take you to it as well. So it just remains for me to say a great big thank you to a lot of different people. I have more slides after this if there's an appetite for them in the questions regarding what each of my students is doing. But I must say a great big thank you to my great mentor, Stephen Gillespie, retired now from the University of St Andrews, but taught me almost everything I know about diagnostics in medicine, and I have a lot to thank him for. Dr. Marianne Abiela is a PhD student in my lab. She's a doctor in her own right and comes to do a PhD with me. Stuart Reed, Helen Onyango, and Joanna Rivas are all working with me as well as PhD students. As I said, I got the prize and Longitude Prize there. IMI have also funded us, as have the European Union, as have the University of St Andrews, where I currently work. We got a prize from the Royal Academy of Engineering, as was said at the beginning. Scottish Life Sciences Award, Longitude Prize Award. We were runners-up at the THE Awards, Times Higher Education Awards in 2017, and Scottish Enterprise helped us with some initial seed funding early on. So I will pause there because I realise I'm at my half-an-hour time limit. However, I have more slides if there's any appetite for them, but at that point I will just say thank you very much for your attention. I hope I've been interesting, and any questions, please.
Thank you so much, Robert. That's brilliant. It's really interesting. Could you tell us what's next for your SLIC technology? I think did you mention, we know you've started your own company, but what would you do differently now and then? Would you just talk us through your potential next steps?
Yes, of course. So next for the technology would be, as you've alluded to, commercialization. So taking it from a box, you saw the picture earlier on, that I build in my lab. They're pretty robust, but they're not very kind of sleek and sexy looking. I'd rather that we had a technology that was completely robust, could be moved around the world very easily, or could be in fact installed in a laboratory like this. You can probably see just over my shoulder, there's a large bio cabinet there. Something about that sort of size that could just live in a hospital lab full time would be great. But really it's commercialization, it's spinning out people with greater engineering knowledge than myself, or knowledge of how to create products for individuals to actually use, end users to actually use, would be fantastic. What would I do differently spinning out a company? I would make sure that everybody involved understood the value of the technology, understanding exactly what it is that it can do. Stories like the one I shared with you all today from the surgical operating theatre is really exciting, really, like I said, first in class, and that sort of thing really needs to be understood and valued by the individuals working with you, working with anybody who's thinking of being an entrepreneur and spinning out their company or spinning out their idea, and understanding the importance and what the machines or what the idea, what the technique, what the assay, whatever it is, what it could do in the future and how important it might be to improve life on our little blue marble.
I mean, from a personal point of view, I think we mentioned before, didn't we, before we came on live, both my brothers are orthopedic surgeons, and for them, antimicrobial resistance is the one global health emergency that actually keeps them awake at night. And this to me seems to be a solution that could solve a problem. But have you ever taken a sample from someone in surgery or through any of your tests which is resistant to all available antibiotics?
Not in my experience, I haven't seen that. I know exists lots of papers and news reports occasionally come out with things like totally resistant. There was a big news report a few years ago about totally resistant gonorrhea. No treatment was available there. I have never seen one that's totally resistant. I've seen some that's pretty close. One of our last line antibiotics as medical scientists is a substance called colistin. And colistin is very, very toxic, very, very nasty, but it's an incredibly potent antibiotic. And I have seen an organism that was resistant to everything but colistin. That patient was treated, they got better. But if that infection reoccurs in that patient or they pass it to somebody else, it may well now have colistin resistance and be completely untreatable, which is a frightening thought. But that's becoming more and more common as we move towards what gets called this antibiotic apocalypse, because we're not undergoing a really important procedure called antibiotic stewardship. We have to look after the antibiotics we have. Don't prescribe them willy-nilly. We have to look after them, keep them, and make sure they're used effectively.
Yeah. So you've talked about communicating the sort of efficacy of your technology when you're spinning out a company, yes. But what else, in your opinion, are the greatest barriers to new health technologies reaching the market?
So some of the biggest barriers are what I've said there. It's taking the idea from a scientist and trying to describe it to someone who's not necessarily well versed in the biological sciences or whatever your area is, if it's in chemistry or physics or a different discipline, psychology, geography, trying to explain the value and the worth of that to somebody who's not necessarily well versed in that area. That can be a major stumbling block because, like I said, the value of it's not understood. Other problems are that the funding available to get these sorts of ideas off the ground are either very, very difficult to write for. They're very difficult to do unless you're very, very well skilled at writing for grants. If someone is not inside a university environment, I would say it's nearly impossible because you need to often have the backing of an institution. And lots of people have fantastic ideas, you know, at their kitchen table or in their garage, and they might even build a small prototype of something, but they don't have the force, the backing of a university or another institution to get them to the funding stage. And it does cost an awful lot of money to try and take something from idea or simple prototype right the way through to commercialized device. There are things called technology readiness levels, TLRs, and TLR 1 is an idea or concept in someone's mind. TLR 9 is a finished, polished product like an iPhone.
Yeah.
And a lot of people sit around TLR 4 and 5 forever and never get any further because they simply can't get the money or the push behind it to get them to commercialization to really, again, help people.
So, a question. This is a very crowded market with multiple startups who've already received funding. How far are you planning to take your project through and I suppose you've slightly answered it, through your company or licensing to an established diagnostics player, which is also an interesting idea?
Yes, so we spun out a company in 2018. Excuse me, in 2018. It started work properly in 2019. And that's exactly what we did. This is when I say we, I mean University of St Andrews. We licensed out the technology to a newly formed company, but that company had the backing of a much larger global corporate. And that's exactly what we did do. Unfortunately, due to various factors, that company folded last year. So there's a whole variety of issues happening there, but that basically means that SLIC in its current form and myself with the IP are looking for intellectual property, are looking for new commercial avenues So in answer to the question of what would I do or what do I want to do, I still want to commercialize. I think the technology has a real opportunity to change antibiotic stewardship and health sciences for the better. So that's what I'd like to do. I'd still like to spin out or indeed speak to a already established company who have a track record who might be interested in working with me.
Yeah. So a technical question, excuse my reading of it. Is your test ISO 17025 or 16095 accredited with traceability to SI units?
No. We have. Thank you for the question. I do recognise what it means. We have not gone through any accreditation processes except the MHRA. So the MHRA was necessary to get us to go to clinics, to go and do our urine and our blood samples, blood studies, I should say. But the FDA and CE marking and all the other ones that come with that, we haven't gone through that process yet. No, it requires, unfortunately, more time than I have to give to it with all my other duties at the university.
Yes. OK, so a question. The kit needs to be rugged enough, particularly you talked about lower income countries, to survive use by non-techies. How sensitive is it to the environment?
Not particularly. I mean, I would have to pick up my computer and sort of point it at it to show you, because I do have one right next to me. But it is a great big plastic box made with metal frame around it and a metal bottom. It weighs about 7 or 8 kilos. And it in itself is pretty robust. It is a laser system, and so a lot of people think, of course, of alignment issues. So the laser might get slightly bumped and goes off alignment and stops working. I thought of that too, and I realized that was going to be a problem. So everything is fiber coupled. So it's all fiber optics coupled directly into the actual device. So where the bacteria sit and where they grow, the light always crosses that pathway in exactly the same place. Every single test. And I must say, well, something I can do is pick up this to show you. I brought this machine to Germany on an aeroplane in this box. And it's a big, strong box, but that's all I did. I wheeled it behind me all the way through the streets of Bonn, where I am, and all the way through the streets of Edinburgh, where I got my flight. And it arrived in perfect working order. So it's pretty robust as it is. But as I've alluded to, working with other companies to get a kind of end user device that's even more robust that you could drop and it would be completely fine. Not something I'd like to do right now. Would be great. That would be the ideal. That's the end result.
So this is an interesting question, particularly when, you know, in medical school we have to consider health economics. They say, is this likely to have a primary care application? We have previously had CRP machines that have been removed as there's been little cost or prescribing benefits.
Yes, so I do see SLIC as a primary care machine. It has applications in both primary, secondary, and tertiary care. So where a patient walks into a GP surgery and says, "Doctor, my foot hurts," and they have a look and it looks like they have some sort of infection, serious or otherwise, take a sample, pop it into a machine, not necessarily the current version, because as I spoke about, I was speaking to you actually earlier on before, but it takes a little bit of hands-on time just now because it's still a research machine, really. In a future version, hopefully they're able to take a swab from a wound or a little bit of blood and pop it into the machine, and it cleverly moves the fluid along a pathway and gets you your result. Even if it did have to use hands-on time, not necessarily for the GP in the surgery or a pharmacist in a pharmacy, wouldn't necessarily have the time to do that. Might have to hire someone else to do it for them. In the current state of play with the machine as it is, that could work. That would be an excellent primary care case. The other applications would be, again, with more development and in a future version. There are machines in labs all around the world, including here. There's actually just one over there, which are great big machines for things like blood culture. And blood is taken by a doctor, or phlebotomist. It's sent to the lab. It's then injected into a small bottle, a blood culture bottle, goes into the machine, and the machine reads it and tells you when it's gone positive. That takes a day or more. exact same thing where we have a special receptacle for blood or urine or any other CSF, cerebrospinal fluid, joint fluid, as I'm doing here in Germany. And you simply load it in and leave it alone. But instead of 20 hours later, 20 minutes later, it gives you a result and tells you, yes, there's definitely bugs growing in there. And then you can go on and do your further testing and do your AST testing if you wished. So there's a use case for SLIC in, I believe, all stages of healthcare.
So interesting. So another question, where you mentioned the interesting part that physics plays in it, but the question is, can you think of any other disciplines or discipline which would have helped you to speed up your research?
Really interesting question. Thank you for that question. That's wonderful. Yes, I am not a physicist. I'm not a materials scientist. I'm not really even an engineer. I got a lovely prize from the Academy of Engineering. That was very kind of them. I suppose you could sort of call me a biomedical engineer, kind of, but I'm a microbiologist by training. That's what I am. And so biology, medicine is my home. That's where I live. So had I had a greater grounding in engineering, in physics, in material science, the ones I've alluded to, but also things like chemistry, understanding the chemistry of the antibiotics I'm using and their interactions with the media in which we grow the bugs, the interactions between the drugs and the media and the sample you're using, even the chemistry or the mammalian biology. As a microbiologist, my mammalian biology, human biology is not as good as doctors and people who study that for a living. Understanding the mixture of immune cells, so immunology would have been great as well. And obviously to say, I would like to have all that knowledge. Of course I would, that would be fantastic, but that's not feasible. So having a team. And one of the most important things I haven't touched upon yet this evening, so I will now, is the most important thing, I think, for success of any project, irrespective if it's an entrepreneurial venture or if it's simply just trying to move science or move another field forward, is having a team around you, a supportive team, all of whom you value and they can value you, that you understand each other, you support each other, and you all have different and varying specialities, disciplines, things that you are good at, and things that you can admit that you're poor at, where you can go to other members of your team and they will help you. So to say, long answer to your question, what other areas would I like to have had expertise in or had expertise around me? All of those different disciplines. I was extremely lucky, and one of the reasons that SLIC was so successful and has been so successful and continues to be is because I actually had all of those people around me. I was able to, because of the University of St Andrews, small place, and the North Hall, where the medical building is where I work, is connected to physics and chemistry and computer science. I was able to simply walk out the door and walk and see my colleagues, and they were able to give me all the help that I needed, which was wonderful.
Thank you. So there's a question here about she says, do you feel you're pushing a big rock up a lonely hill, or is the specialty community worldwide big? Is humanity basically making good progress here? Are you optimistic that we will act in time? So
the question is this the Sisyphean task? Am I pushing a rock up a
yes.
hill? I don't believe so. I think that antibiotic resistance is never going to go away. The bacteria will not stop evolving. And we're not about to eradicate every single one of them off the surface of the earth, because that would be absolutely awful. It would kill everything else. See people who unfortunately need to have their bodies sterilized due to having various medical conditions, and when if they're ever able to re-enter the world, which is rare, horrible skin infections and things because they've had to be recolonized by organisms. So killing all the organisms is not a good idea. It's not even something worth thinking about. So instead, we have to consider what we're going to do to help us coexist with these organisms that we've been living alongside, or life on Earth has been living alongside them for billions of years. And we, for the last 100 years, have been throwing incredibly high dose chemistry at them, and they have been reacting in a evolutionary fashion to protect themselves, as any life form would. So technology like SLIC, things like it, other technologies that will be available in the future that will allow us to, instead of going through empirical therapy, as I alluded to earlier on, to make a clearer point of what it is, empirical therapy is guesswork. It's saying there's an infection in the shoulder joint, I will prescribe this drug because that's what the pathway tells me to do. Instead, we wait for rational therapy. Wait just a short time with technologies like SLIC and we can get the correct result, as it were, and prescribe properly. Things like that are really going to help. Other work that I'm doing, projects with my colleagues. So Mariana Abela on there, on that last slide that's still showing, is working on PDT, photodynamic therapy, a way of killing organisms using light and another chemical that doesn't involve antibiotics at all, and it kills the organisms so quickly they have no chance to develop resistance. So other means of killing the organism when they're causing an infection is really important. Other work I'm doing with colleagues is working on combination therapy. Instead of using a single massive dose of an antibiotic, we use a very, very tiny dose of multiple antibiotics. And that means the chances of them evolving resistance to that are very, very small. It rises exponentially with each new added antibiotic. So instead of turning up to the doctors very, very unwell and getting a massive dose of meropenem into your veins through an IV, you would instead get a very tiny dose of meropenem plus gentamicin plus cefoxitin plus ampicillin plus amoxicillin, perhaps. And that combination might be an amazingly synergistic combination that kills the organism that's hurting you. How did you find out what it was? You used SLIC to begin with. So it's not a Sisyphean task if we realize, and when I say we, I mean the scientific community, but actually more importantly, policymakers recognize the importance of what is happening, of the antibiotic apocalypse as it gets called, and do something about it. And the people on the screen here, the individuals, Joanna, Helen, Stuart, Mariana, Stephen and myself, but also groups, Nesta, who run Longitude Prize, Life Sciences Award, the Times Higher Education, European Union. There are groups who are recognizing this issue. They just need to push harder for longer, and we as scientists need to encourage young people, more young people, to come into this area and research it, because not only is it fascinating and great fun, it's also incredibly important.
Thank you. Have any of the old-fashioned sulfonamides been re-evaluated? Does that make sense?
It's a very, very niche question.
Yeah.
Have they been reevaluated generally in the world? Possibly. I don't know. I've not read every paper out there, unfortunately. Have I looked at sulfonamides on my testing rig? Yes, I have. They are somewhat effective, but generally speaking, there is a lot of resistance to them out there. Short answer.
Brilliant. Okay, well, thanks so much, Robert. I have one last thing, a lovely post. Somebody saying it would be great to see you succeed. They helped with the success of Axis Shield across the Tay, but they're rooting for more for a St Andrews player. So we'll be in touch. I just want to...
Very, very kind of you.
Yeah, thank you so much, everybody, for coming. Thank you, Robert, for your great talk. Our next Saints talk is Professor Len Thomas. That is Tuesday, the 23rd of May, and the title of his talk is Counting Critters, St Andrews Research at the Interface Between Statistics and Global Wildlife Conservation. Just want to say a huge thank you to everybody for coming tonight.
Thank you very much.
Thanks, Robert.